Okay, hi everyone. My name is Sayani Banerjee. I'm a PhD student at the Department of Developmental Cognitive Psychology at the University of Regensburg, and I will be talking about how we explore the neural foundations of infant motor development and spatial cognition. We focus on motor development because infant motor skills develop quite rapidly over the first 2 years of life. And especially in gross motor skills, we see that infants go through significant milestones, like the transition from crawling to walking. And in terms of neural development, we can assume that there's a lot of changes going on in the brain during this transitional phase. We also have a lot of evidence over the last decades that infant gross motor skills and fine motor skills have a significant impact on a lot of cognitive skills that develop around this age. Not just in the spatial cognition domain, but also in social cognition domain. But for this study, we mainly focus on spatial cognition. So for this study, we invited infants to test if we can replicate the association between early gross motor skills and spatial cognition, and more importantly, if we can characterize the neural underpinnings of this association. So we invited infants at 12 months of age to test their gross motor skills and spatial cognition. And for this behavioral sample, we have 108 subjects. And then we invited a subsample of them for an MRI scan at 13 months. And for this, we have 41 subjects with complete successful scans. And this reduced sample is because of how difficult infant MRI is, especially if you're doing resting state MRI, which is when you scan them when they're naturally sleeping. So one of the main challenges of our study is how we can interpret neural underpinnings with small sample sizes. Before I go into further details about this, I would briefly talk about how we test spatial cognition in these infants, and we do that with the help of two tasks. So the first one is a task adapted from the study by Dahl and colleagues from 2013. We perform this in a virtual reality environment called the cave, and here there are 5 projectors projecting on the 4 walls and the floor, and we test infants' self-body movement as a response or as a compensation to peripheral optic flow. So this is what it looks like. And the other spatial cognition measure that we test is spatial navigation. This is a task adapted from the Clearfield study from 2004. And here the infant sees the parent hide behind one of these 3 walls. And then the infant is disoriented at the center and then also distracted with either a cloth or a toy so that they don't fixate on the wall that they saw the parent go behind. And then we ask the kid to locate the parent.
Wo könnte sie sein? Sollen wir mal gucken?
Hallo! Hallo!
So in our behavioral sample, we found that infant gross motor skills were significantly correlated with the visual proprioception measure, which is the moving walls task. And this correlation was also significant in our smaller sample for the MRI subjects. And then similarly, the gross motor skills were also correlated in the behavioral sample with our spatial navigation measure, but then not significantly correlated in the smaller MRI sample. So this tells us that the visual proprioception measure seems to be a bit more robust, and comparatively, the spatial navigation measure is not as sensitive when we look at smaller sample sizes. So coming back to our research question, we mainly wanted to find out how we can understand the neural underpinnings of this motor perception link in small sample sizes like ours. And to make any kind of brain behavior associations, it is quite difficult to do with the kind of sample size that we have. And one of the approaches that we could potentially use is normative brain charts. So this is an example of a brain chart from Bethlehem and colleagues from 2022. They used over 1,000 data sets to chart the human brain across the lifespan, so from prenatal until old age. And then you can see for gray matter volume and white matter volume the metrics, they are increasing quite rapidly from birth up until early childhood, and then it slowly decreases towards old age. Now, when we focus on the infancy age range, there are a few challenges that come up. Especially in these lifespan models, infants are heavily undersampled, so these kind of models are not as precise. And till date, we don't have any infant brain charts that will allow us to chart the human brain specifically within the infancy range and also allow us to make any meaningful associations with behavior. So we created infant brain charts focused on this age span of birth to 2 years using the Baby Connectome Project data, or the BCP. The BCP gives us over 600 anatomical data sets that we can use, and we pre-processed and segmented the BCP data set as well as our smaller MRI sample to extract cortical and subcortical regional volumes across the brain. And the next chart for creating of the brain charts was modeling. So we fitted a general additive mixed model, or a GAM function, on the regional volumes as well as the whole brain volumes across the months. And here you can see on the screen the GAM fitted on the whole brain, so the ICV, or the intracranial volume, from birth to 2 years. And as you can see, the volume is increasing quite rapidly over the first 2 years, which is also what we saw in the lifespan models. And then the next step is more crucial, which is normalization. So as you can see, the whole brain is developing quite rapidly over the first 2 years, and we wanted to see how individual regions are developing with respect to the other parts of the brain. So we divided the growth function fitted on individual regions with the growth function fitted on the ICV, or the intracranial volume, and then we found very distinct developmental patterns for individual regions, which told us about the relative growth of every region when controlled for the whole brain. So for example, here you can see on the left, the hippocampus and the precentral gyrus. Before normalization, they both show an upward trend. But then after normalization, you can see that the hippocampus shows a downward trend, which means that it is developing slower compared to the rest of the brain. And then the precentral gyrus shows a slightly upward trend, which means that it is growing faster than the rest of the brain until about 12 months. And then its growth rate sort of aligns with the rest of the brain. And after these infant brain charts, then we can extract different neural metrics. In our larger BCP sample, we extracted a brain age measure to sort of validate the charting approach. And here we used the brain age to sort of characterize the global neural maturity. And this was done with the help of normalized residuals. So normalized residual is the difference of the observed normalized volume of a region and the normalized volume predicted by the brain chart for individual regions. And then we estimated the brain age by the minimal sum of squared residuals across all the regions. And then we found that this brain age measure in an age-corrected analysis was significantly correlated with cognitive development in the BCP data, which means that already in our larger sample, we were able to find a significant association between the global neural maturity and cognitive development. So that was great. However, we found that this brain age measure is not as predictive when we look at smaller groups of regions. And because we are interested in motor development, we wanted to focus also on regions that are related to motor skills. So we focused on certain regions that we know from human adult studies or animal studies are relevant to motor skills. And these are the precentral and the postcentral gyri, which are said to be part of the sensory motor cortex, and also the hippocampus and the entorhinal cortex, because they are related to spatial cognition. And then we combined these regions to create certain neural metrics in our smaller sample, which is what you see overlaid here in red on the ICV chart that we derived from the bigger data. So the neural metrics that we derived, firstly, was the normalized residual, so same as the BCP. And this gave us, for each region, the deviation of that subject from the normative brain chart. And then we created a neural measure by combining or summing up the residuals across all the selected regions. And then another really informative metric that we extracted was the slope. So the slope gives us the direction of the developmental pattern at any given time point. So if the slope is positive, then it means that this region is developing faster than the rest of the brain at that given time. And if the slope is negative, it means that this region is developing slower than the rest of the brain. And you can see after normalization, different regions have very distinct developmental patterns when you control for whole brain growth. So the slope metric helps us to capture these sort of unique differences in the developmental charts. And then because the residual and the slope are equally quite informative, we wanted to integrate them into specific scores. So we created the product for every region of the residual and the slope. So this gives us basically the position of a subject for each region at the normative brain chart. So it takes into account not just the deviation from the normative brain chart, but also the slope or the growth pattern for each region. And then we also summed this across our selected regions to create a neural measure. The other neural measure that we created was the overall correlation between the residuals and the slopes across these selected regions. So this gives us basically the correspondence between the deviations from the norm for that subject and the actual growth patterns of these regions. And then we also derived a latent variable, which was derived from the residual, the product, and the slope. So in the end, we had 4 different metrics. Now, before we applied it in our smaller sample, we wanted to see if it does, if it is associated with gross motor development, which is what we are interested in the larger sample, so the BCP data set. And then, excitingly, when we focus actually on the age range where the infants are transitioning from crawling to walking, we found that all of our neural measures, except this correlation measure, was significantly correlated with the gross motor score, which is amazing. And then we wanted to look at the smaller sample, so because we want to, in the end, use these infant brain charts to correlate it or to see its association with motor skills in the smaller sample. So amazingly, we found that it does correlate also with the gross motor skills in the smaller sample, other than the correlation measure, which is also what we saw in the BCP data set. But that is not it. We wanted to see if it also correlated with the spatial cognition measures that we have from our behavioral samples. So when we look at its correlations with the behavioral measures, none of the metrics were significantly correlated. But then we did see that these neural metrics are strongly correlated with the visual proprioception, but then the significance is, it's very close to significance. And then this correlation measure was, in one-sided significance, correlated with the visual proprioception measure. After this, we wanted to see if, so which are the regions within this selected group that is sort of driving this association between gross motor skills and the neural measures that we've extracted. So here, we correlated the residuals combined across hemispheres of all these selected regions, and then correlated it with our gross motor score. And here we found that the hippocampus residuals correlate with the gross motor scores. And that is also very interesting. But then we also correlated it with our spatial cognition measures. And here, none of the correlations were significant. However, quite interestingly, the residuals of the precentral cortex and the postcentral cortex were correlated with one-sided significance with the visual proprioception measure. So with these results, I would like to summarize our findings. So we were successfully able to replicate the previous findings of Dahl and colleagues and also Clearfield that early gross motor skills are significantly associated with visual proprioception and spatial navigation. And in the BCP data set, we found significant associations between neural maturity and not just cognitive development, but also gross motor development. And then, which is also what our sort of main goal of the study was, we were able to use infant brain charts based on big data to characterize the neural underpinnings of infant gross motor development in a smaller sample like ours. However, because of really low sample sizes, we were not yet able to find a mediation link of neural maturity in this association between infant gross motor skills and spatial cognition. However, we did see that the hippocampus seems to be quite relevant for infant gross motor development. And for that, we went another step in our segmentation, and then we segmented the hippocampus into the hippocampal subfields using a different segmentation pipeline. And then we found, so we got the volumes of the hippocampal subfields, and then we're using it right now to further investigate the hippocampal maturity, and then it's linked to not just spatial cognition, but also gross motor development. And with this, I would like to end my talk, thanking everyone for your attention, and also the members of our lab and our collaborators in Berlin, and also at our university in Regensburg. And of course, everybody, all the families who participated in our study. Thank you very much.